So today we are going to discuss number 4 to number 6 of chapter 6 workbook lo. Okay, let's see number 4. A student carried out an experiment to investigate the effect of light intensity on the rate of photosynthesis. So here you should know that the light intensity is the factor that we want. So this one you can say it as the manipulating factor. And we call it as MV lo. Not factor, sorry, variable. Okay, while the rate of photosynthesis is the responding variable. Responding variable is the one that we the effect lah, the results. The student changed the light intensity by moving the lamp at different distance. So this is the way that they do lah for the manipulating variable, different distance, away from the beaker containing pond wheat. Okay. Stopwatch is over here, thermometer over here, beaker with distilled water, and then pond wheat, water with sodium hydrogen carbonate, lamp and ruler. Sodium hydrogen carbonate we already know this one is to supply carbon dioxide. Okay, let's look at A. State 2 ways to ensure that the temperature has no effect on the result of experiment. Okay, they don't want the temperature to be one of the manipulating variable. So this one, the other way that they ask this question is how to fix the temperature of the experiment. Okay, so we have several ways lah. So this one, the first way is notice that the beaker here is filled with water. Inside here also got water, but outside here, why do we have to put another how to say a layer of water, like water bath like that? Why? This one is to make sure that the temperature won't be changing lah. So no matter how the temperature outside here is changing, increasing or decreasing, because of this water, the temperature inside the test tube it still remains the same. So this one we can see place sorry, this one is we already have the beaker over here. We add one more glass tank over here, which is filled with water. So this one even though the light here, the light intensity increase or decrease, the temperature still will be the same lo. So glass tank filled with water between beaker and lamp. So this one, the word that you must write, the phrase that you must write lah, glass tank filled with water. Must be water lah, cannot be oil lah or sand lah. Okay. The other way is thermometer. So you can say, use a thermometer. And then state the function. To monitor the changes of temperature. Okay. Thermometer, monitor changes. Okay, so this is for 4A lah. Let's see 4B. The student obtained the result by counting numbers of bubble release by the pond width within a minute. State the hypothesis. Okay. Hypothesis is easy lah. You can say the greater or smaller, faster or slower. The MB, the whatever, the RV. MV is manipulating variable. Here already know lah. This one is MV. Different distance. This one is the responding variable. So just write lor. The shorter the distance between the lamp and the beaker, the greater the number of air bubbles released by pond width within a minute. Okay. C. State one more accurate way of measuring the gas given off by pond width in the experiment. So this one you can say that instead of counting the number of bubbles, you can choose to say use a string lor, with readings to determine the volume. Okay, so this one is more to like a experiment questions lah, but this one is quite easy lah, not hard lah. Okay, and this one we should not use air, this one we should use gas. Okay. So 1 mark over here. 1 mark over here. And then this one, for B, 1 mark is shorter and then NV. Greater and then RV. Okay. Let's look at number 5. Okay. The leaves needs to be distarched before they can test it for presence of starch using an iodine solution. So, iodine solution, if starch is present, the color will go from yellow to blue-black, or you can say dark blue. If starch is not present, the yellow color will remain unchanged lah. Okay, so this one is the starch. The starch basically means removing starch lah. Okay, you can see that the water over here, they heat, or you can say they cook the leaves lah, and then using Bunsen burner, and then after that they put it inside a water bath. They put the leaf into a ethanol and then put the test tube inside the boiling tube. Put the boiling tube inside the ethanol. Water bath, this one. And then after that, by this time, after that they put the leaf into the distilled water, and then only they test for iodine. Okay, this one, based on figure 5.1, complete table 5.1 by identifying the purpose of carrying out each step in the process of distarching. Okay, let's see. The first one, keep the leaf in a dark room for 48 hours. So this one is dark room. If dark room means no photosynthesis can occur lah. So this one you can say that to allow to make sure or ensure all starch stored in chloroplast is used up. Boil the leaf for 30 seconds. This one is to break down the cell membrane. Okay, this is because the leaf usually they will be boiled for 30 seconds lah. And then to kill the cells in leaves and break down cell membrane. Okay. Okay, so this part is to actually allows the iodine solution to get through the cell membrane, because cell is like that one, plant cells. This one we take the palisade mesophyll cells lah. Sorry, not middle. Something like that lah. Okay. This one, they break the cell membrane already, and then after that, only the iodine solution can pass through the cell membrane and then move into the chloroplast and then reacts with the starch ma. So this is why we have to boil the leaf lah, and then heat the leaf in ethanol. This one is to remove chloroplast sorry, not chloroplast, chlorophyll in the leaf. This one because normally when we see the leaf is in green color one ma. So when we test iodine we need to know that if there is starch. If there is no starch we have to put yellow color ma, right? And then if the leaf is green color, then you cannot see the color, you cannot see the results clearly. So they need to remove the chlorophyll first lor. Okay. And then heating the ethanol is to, this one is related to chemistry lah, is to increase the rate of reaction. Okay. Dip the leaf in distilled water. This step lor. This is to soften the leaf. Because we already heat ma. By this time the leaf already becomes can be destroyed very easily. We say it as very brittle. So this one, we rinse it in water lor, so that it won't break easily lah. Okay. The boiling tube containing the leaf and ethanol is heated using a water bath. State why is it important to use water bath. Okay, they ask why. So this one is because ethanol is extremely flammable. And then ethanol can vaporize easily. This one we have another word, we call it as volatile. But if you write this, then you can get your marks already. And then extremely flammable. Okay, so this is how you get the marks for A and B, 4 marks plus 2 marks. And then after that, C, predict the results of iodine test. The starch leaf means starch is removed. Means no starch already lah. So if no starch then the iodine solution remains in, you can say light brown or yellow color also can. Okay, so this is for number 5. Let's look at number 6. Chloroplasts play an important role in carrying out photosynthesis to manufacture carbohydrates and plants. Okay. Figure 6.1 shows a variegated leaf used to investigate the effect of chloroplasts on the photosynthesis of plants. Okay. Variegated actually means that the plants have different parts lah. One part is white color or sometimes it's yellow color or red color or purple also can. While the other one is green. Means that this leaf not the whole leaf is in green color lah. Only some part of the leaf is in green color. Okay. Let's look at the next page. Okay, explain the term variegated leaf. Okay. Oh, sorry the other part is always white color. So this one, write down the term first. And then after that is, and then only you write down the definition. A leaf that is partially green and partially white. While the green part of the leaf contains chlorophylls. Okay, so this one partially green and partially white, one mark, and then the other one green part contains chlorophylls. This one is your second mark. Okay. B. The variegated leaf is tested with iodine solution for the presence of starch. So you have to predict and then explain the result. So what will happen? So let's predict first. Green part means that chlorophyll is present. Means that there is chloroplast lah. Means photosynthesis occurred. Glucose produced means they are starch. This one, white part white part we have no chlorophyll because it's not in green color means no green pigment already lo. And then no chlorophyll means no chloroplasts, means that no photosynthesis occurred, no glucose is produced, no starch is formed. Okay, so by write this part, the iodine solution they will go from yellow or light brown to dark blue or blue black. For this one, the white part, yellow or light brown remains. So let's see the result here. How can we write? the light brown color of iodine solution turns blue-black on the green part, while the light brown color of iodine solution remains unchanged on the white part. Okay, and then after that, so you have 1 mark for turns blue black, and then the other one light brown remains. 1 mark for green part, 1 mark for white part. Okay, explanation why. This one you have 2 marks for explanation. So you can say that photosynthesis occurred or occurs lah, depends on you lah, in the green part of the leaf. In the presence of chlorophylls. Hence, starch is produced and turns the iodine solution into blue-black. Okay, and then after that, you talk about the white part. White part of the leaf has no chlorophyll. Hence, no photosynthesis occur, or you can say photosynthesis does not occur also can lah. And no starch is produced. Therefore, iodine solution remains light brown when starch is absent. Okay, 1 mark is in photosynthesis occurred and then presence of chlorophylls. So starch produced and then turns iodine solution into blue-black. This is your first mark. And then your second mark. White part has no chlorophyll. No photosynthesis occurred. No starch produced. Remains light brown. This is your second mark. So that is all for biology. See you in chemistry.